Hello everyone, this is going to be the first video that's going through the second lines of defense for our innate or non-specific immune responses. So I updated this slide just before making this video, so your PowerPoint may only have 7 little rectangles, but I have updated it to 8. So please make sure to add on the antimicrobial peptides. All right, so remember, we are talking about our second line of defense. And so these are going to be cells and processes that are always on the ready, looking out for foreign invaders. So essentially, if something gets past our first line of defense, like the skin, mucous membranes, etc., then these cells and processes are right there trying to catch things before they get out of hand. And so that's why they're called our second line of defense, because they're only going to be active. Well, they're always active, but they're only going to come into play if something actually makes it inside the body. Okay, so as you can see here, there are going to be 8 different cells and or processes that we're going to talk about. So this video is going to focus on the first 2, interferons and phagocytosis. So in order to understand all of these, before we dive into those details, we're starting to really get into immunology, which is the study of our body's immune response, and specifically our immune system. And so the purpose of our immune system is to constantly be on surveillance. They're constantly looking for pathogens. But not only are they looking for them, they have to be able to deal with them. So they have to recognize something as foreign and target it for destruction. And so key to the success of our immune system are white blood cells, also called leukocytes. And something that's going to be a little bit more pivotal in future videos is going to be this ability of our white blood cells to differentiate between self versus non-self cells, which is just a sciencey way of saying cells that do belong and cells that don't belong. Okay, that way they can identify those pathogens and remove them, hopefully, before our body reaches a diseased state. Okay, and so here's just kind of visualizing that a little bit. So our white blood cells or leukocytes should be able to detect any cell that it comes into contact with and say, hey, you do or don't belong. And so what it's doing is essentially looking at cell surface markers and saying, oh, yep, you're supposed to have those. Good, you belong here. And so then no reaction is going to happen. However, what this same white blood cell or leukocyte can do is when it detects something foreign on a cell that it comes into contact with, it says, oh, no, you should not be here. And then some of them can immediately phagocytose and kind of present that invader to the rest of the immune system. Okay, so we're going to talk a lot more about this later, but that is one key part of our immune system, is that certain cells can differentiate self from non-self. All right, so now we're going to move on to kind of the meat and potatoes of this particular video, which is going to be all about interferons and then the process of phagocytosis. So interferons are little proteins that serve as signaling molecules, one type of signaling molecules, I should say, which cytokines is kind of that fancy word for signaling molecule, but there's tons of different cytokines based on the cell that is releasing the signal, what the signal is. So interferons are one type of cytokines. And interferons have a variety of roles in the cell, as you can see in the picture. We're going to focus on the role that interferons play during a viral infection. Okay, so they do more than that. They are not just for viral infections, but for the purpose of this course and what you need to know for micro, we are going to focus on that. Okay, And so essentially, when a cell is infected with a virus and the virus is replicating, that cell eventually knows something is wrong, right? Because the virus has taken over all of its processes, the cell is going to be getting sick. And so, what the cell essentially does is send out a warning to neighboring uninfected cells. And so it tells those uninfected cells, all right, hey, there's a problem. Okay. You need to kind of temporarily shut down. You need to destroy RNA. You need to stop protein synthesis, right? Because these are things that a virus can manipulate. So you essentially need to just kind of chill for a little bit until we know the threat has passed. These interferons also signal other cells that are also infected to undergo apoptosis, which is programmed cell death. And so essentially, it's saying, hey, you cell are also infected. So you need to essentially take one for the team, and the cell implodes. So that cell dies, but it traps any virus that's in it inside. So the virus cannot get out and cannot infect a new host. The other thing it does is it signals uninfected proteins to create something called APCs. I'm sorry, I just butchered that. AVPs, which stands for antiviral proteins. And you can think of these AVPs as a little military that these uninfected cells make. And these proteins, these AVPs, kind of stand guard and wait for a virus to try to infect. And if a virus does try to infect that cell, the AVPs are able to shut it down so that it can't actually harm the cell. So the last thing it also does is it warns the immune system. It says, hey, there's a problem. There is a virus on the loose. We need to be on the lookout for it. So it's going to activate other immune cells that have a bigger role in our third line of defense. So we'll talk more about them later. But it's activating them so they can start trying to find the virus and hopefully eliminate it before it causes too much damage to the body. Okay? So again, big key take homes here. The interferons are a type of cytokines produced from a virally infected cell to warn uninfected cells that they need to be prepared. They warn or they signal other infected cells to do apoptosis, so they can essentially implode and trap any virus particles inside them, again, trying to limit the spread of this virus. And they also activate immune cells so that our third line of defense is on the prowl trying to find and eliminate this virus. All right, so those are interferons. This next one is a process that you may have heard about, especially if you took anatomy and physiology. And this process is called phagocytosis. And essentially, this process, kind of in more common terms, is where phagocytic cells, which are immune cells, they hunt down, capture, and eliminate pathogens. That's big picture. Find them, catch them, eliminate them. Okay, so here's the 7 steps of phagocytosis with some bold and I guess what is that, orange, redishy orange colored terms. So you do need to know the micro terms for this. Okay, so the first thing that has to happen is the phagocytic cell is going to essentially sniff out and hunt down the pathogen. And it does that using a process called chemotaxis, which is where, let's talk bacteria, for instance. As bacteria are surviving and doing their thing, they just naturally release little waste byproducts. And that provides like a chemical stimulant or kind of a scent, if you will, that the phagocytic cells can detect because it's a bacterial waste product, not something that our body is naturally making. And so the phagocytic cell kind of acts like a hound dog and like sniffs it out until it can find it. Is that step one? It's trying to essentially hunt down the bacteria using a process called chemotaxis. Once the phagocytic cell has detected and is in close proximity to the pathogen, it has to attach to it. It has to essentially grab hold of it. That process is called adherence, right? To adhere means to attach. And so, specifically, the receptors on the phagocytic cell attach to PAMPs on the pathogen. Okay, so PAMPs are just like pathogen associated molecular patterns, basically something that's foreign. So, like just a surface molecule on the pathogen. Okay, so the receptors on the phagocytic cell are going to attach to molecules on the pathogen. So it's like essentially think of the dog has just sniffed down the bacteria in step one, and now it's jumping and it's latching on to that pathogen. Okay. So once that happens, okay, so if you're looking at this image here, right here, do you see how the receptor on the immune cell, the phagocytic cell, has attached to the pathogen? So now what's going to happen is the phagocytic cell, part of that membrane goes inward, okay, because it's trying to essentially grab hold of. It looks like it's got to like just jaws or something coming up and going to bite all of these little pathogens. And so this is called endocytosis, where the cell is taking these bound pathogen inside of the cell. Okay, so that's called ingestion or engulfment. So at this point, once that has happened, now notice that the pathogen is kind of quarantined or jailed, right, in its own little vesicle or vacuole. And that vacuole has a name, and you need to keep track of these names, okay? So notice when it's just the full intact pathogen just hanging out by itself in that little vacuole, we call it the phagosome. Okay, so think of this is phagocytosis, and now it's in a little vacuole, so it's called the phagosome. But notice what's happening in this picture, y'all. Do you see these little lysosomes? It's on their way. Well, it's really just one in this picture. One lysosome is traveling to the phagosome because the purpose of this cell is to eliminate those pathogens. And remember, the lysosome, its main job is digestion. It's going to chop these little pathogens to bits, okay? So if you notice in step 5, the lysosome, full of hydrolytic enzymes, has fused with the phagosome. So now we refer to this vacuole as the phagolysosome. So notice we just merged those names. Phago from the phagosome and lysosome because it's the lysosome. So it's here that the microbe is actually killed. Remember, all those hydrolytic enzymes, their purpose is to chop those pathogens to bits. Now, you want to think back. What did we learn about the lysosome in Unit 1? Remember, those hydrolytic enzymes only work in an acidic environment. So that phagosome in step 4 actually has little proton pumps that start to acidify the phagosome as soon as it forms. That way, when the lysosome comes in and fuses with it, it has that acidic environment required for the hydrolytic enzymes to do their work. So once the hydrolytic enzymes have digested those pathogens, eventually all that's left is indigestible material. And so now the digestion is done. And so this vacuole is called the residual body because it's got the residual pieces of bacteria that couldn't be broken down. And the cell does not need to keep that. It's all waste. And so the last step of this process is exocytosis, where it essentially just dumps all that stuff outside of the cell. Okay, so I'm going to go through that a little bit quicker. I'm going to do it in common terms, and then I'm going to do it one more time using scientific terms. So essentially, step one, the phagocytic cell is going to sniff out and hunt down the pathogen. Once it finds it, it's going to grab hold of it. And I think of ingestion like somebody catching somebody, like tackling them, like in a big bear hug. That's going to be where they're kind of ingesting them. Okay. Now think of it as that thief, that pathogen is now in jail. Okay. And now we're going to, introduce the digestive lysosome here. That pathogen is going to get, essentially chopped to bits, and all the leftover parts are going to get removed from the cell. Okay. So again, sniff it out, hunt them down, catch them, isolate them, chop them up, throw away the excess. That's essentially what just happened. Now, using sciencey terms, the phagocytic cell hunts down the pathogen using chemotaxis. It is then going to have its receptors bind to surface molecules on the pathogen. The phagocytic cell then endocytoses the pathogen, bringing it inside. There, the pathogens are kind of quarantined in a little vacuole called the phagosome. The phagosome has proton pumps, which are slowly kind of acidifying that environment. That way, when the lysosome arrives and fuses with the phagosome, okay, the environment is compatible for those hydrolytic enzymes. Once that fusion has occurred, we now refer to that vacuole as the phagolysosome. It is here that the pathogen is exposed to the hydrolytic enzymes, which digest that pathogen. Once digestion is complete, all of the indigestible parts are left. The vacuole is now called the residual body, and those indigestible pieces are exocytosed from the cell. So notice, y'all, there is a little animation if you want to check it out. I always suggest seeing an animated process. It kind of helps these terms and steps come to life. All right, y'all, that is it for this video. Please let me know if you have any questions.